for neurobiology and brain restoration. We aim to, firstly, to establish communication between specialists in different areas of neuroscience and to share knowledge with the general public and everyone who is interested. Today we talk about neurogenetics, and we have a guest. It's Oleksandr Frei from Norwegian Center Neuroimaging for Mental Disorders Research. He will talk about a statistical approach in neuroimaging and psychiatric genetics. So,
thank you so much for kind introduction, and it's really a pleasure to present here at Scotech. So today I represent Norwegian Center of Mental Disorders Research. My name is Oleksandr. Originally I graduated from MIT here at Dolgoprudny. So that's my background, more technical computer science and applied mathematics. But today we have a kind of long list of topics to go through, so I'll spend some time on this slide so you guys have an overview of what we'll be talking in which order. So I'll start by giving a brief introduction. Why do we at Neuromed study psychiatric disorders? Why is that important for society? Also, I will discuss what data do we have available to us and how do we organize our work and groups in Neuromed, and particularly present the research by my colleagues in the multimodal imaging group, because it's of special relevance here at CNBR. And then we will sort of move into my favorite area of research interests in genetics of complex human traits. So the second part will be kind of 101 general introduction, kind of educational day on the genetics of complex traits. And then we move, maybe after a break, we'll see how do we feel after, to the really advances in statistical analysis. So, yeah, so starting here, why do we study psychiatry? Because these disorders, the mental disorders, have high cost, both in terms of just money for society and also a huge emotional burden for individuals and their families. So from ethical perspective, it's very important to study. The psychiatric disorders have pretty much unclear etiology, which means that we don't know exactly what's going on wrong, what's wrong. Like it's not an influenza when we know that people get caught some virus, right? It's really unclear what's going on, and it's unclear in terms of prognosis when they arrive at clinics, what will happen 10 years later. The best treatments we have so far have lots of side effects, and also some of them don't work at all, so that's what we mean by treatment resistant. There are large class of comorbid disorders which happen together with schizophrenia, including cardiovascular disorders and many others, and even mental disorders are frequently comorbid with each other. Surprisingly, psychiatric disorders are very common. I think that depression or major depression is probably 1 out of 6 people will be diagnosed with this disorder at some point, and schizophrenia is like in the order of 0.5% or 1% prevalence, depending on how you count. So this is a very, very common disorders, in addition to the issues I've highlighted above. So yes, naturally, mental disorders are a very major public health concern, and the cost, even like financial cost, including the potential financial loss from people not being as productive due to mental health, is far in psychiatric conditions far outweighs the cancer, for example, which is, I think, normally not something general public is aware of. So therefore, the development of novel treatments, both drugs and some kind of prevention strategies, is really high on the list of European research. In addition, so we as society would like to, the tax to be distributed equally among everyone, but the psychiatric patients didn't benefit as much as the rest of the population from the decades of advances in different areas of medicine. So the gap in life expectancy, for example, still keeps increasing rather than sort of being more fair across different groups. So here I'm specifically talking about schizophrenia because this is one of the main traits we work with in the center. And to treat schizophrenia, we have a large class of drugs called antipsychotic medication, and they're generally regarded as effective drugs. However, normally when people think of schizophrenia, we probably imagine people hearing voices or seeing things like hallucinations, but there is a whole other dimension to this disorder, which is about negative symptoms. So people affected by schizophrenia also feel maybe a lack of empathy, or it's hard for them to connect with other people. They might experience mild to severe cognitive dysfunction, like memory issues, etc. And this kind of whole domain of symptoms is not really well treated by the antipsychotic drugs. They might kind of help in the first episode psychosis to get rid of psychotic issues, but not this negative symptoms class. And in addition, these are really heavy drugs that cause a lot of side effects. So weight gain, dyslipidemia, shaking hands, and that kind of forces many people to quit from treatment even when it's effective. And in addition, from 30 to 50% of the patients, they don't respond to the treatment at all. So they might still get the side effects that I mentioned, but kind of they have to try another drug and then get another drug, and it kind of doesn't get an improvement that was promised to them. So largely that is due to the fact that the antipsychotics we use today were discovered was the first class antipsychotics was discovered almost like in 1950s. And 7 decades later, that's more or less still what we have. So of course there is some progress, but it's not nearly as large as in cancer or cardiovascular or many other domains in the medicine. So even though we know, the mechanistic way how the drugs work, it's not necessarily the way they help us to cure schizophrenia. So that relation between the drug and schizophrenia remains unclear, and that is one of the reasons why it's hard to develop novel drugs that are more effective. And that's why there is a growing interest in genetics research for schizophrenia, because this schizophrenia is a highly heritable trait, one among the most heritable psychiatric conditions, and therefore we hope that by studying genes associated with schizophrenia, we might get some hints where to search for better drugs. Kind of moving a little bit back towards comorbidities, schizophrenia, bipolar, and major depression disorders have significantly higher mortality rates in the general population, and this is to a large extent due to cardiovascular risk factors. So, on average, people affected by mental disorders have 15 to 20 years shorter life expectancy. It's quite a serious issue, and this is mainly due to threefold increased risk from dying by cardiovascular risk factors. So, and even though we've known these issues like for decades now, the gap is still there. There was nothing we could sort of help to resolve these issues. And therefore, at the moment, we have these two major aims about better understanding of the mechanisms behind psychiatric disorders, based on our recent discoveries of genetic risk factors, and develop tools for stratification of the patients into groups and better prediction of the outcome, so that therefore we can do translational studies and bring that back to clinics. So the four further aims are sort of sub-goals, more specifically what we are working on. It's about disclosing the genetic architecture, both in terms of rare and common variants, and the pleiotropic variants, meaning the relationship between genetics across traits. And we also work to identify novel brain phenotypes, features in the brain that would be intermediate phenotype between the genetics and the clinical symptoms. And those are two about the second aim, about statistical tools, using genetic, environmental, and clinical factors to predict prognosis and outcome, and eventually to translate it into clinics. So the center is organized into these 7 big core resource units, and each unit is led by a principal investigator and a group of between 10 and 15 other personnel, mainly PhDs and postdocs. And the key idea is that these different units will not sort of work in isolations, but we will design a project that spans across these projects, and we will also give access to our benefit from the expertise from our collaborators and plug them into the specific projects so we can have some synergy. So moving more towards the data that we have access to us to tackle these problems is around 120,000 of Norwegian samples that we currently have in-house with full genome-wide genetics data. It's of course, it's not a whole sequencing data, that would be a lot, but it's a genotype data that we have available with phenotypic data through Norwegian registries, including like years of schooling for everyone, the clinical history, so the registry data on diagnosis, etc. Basically everything that Norwegian government collects about people we could potentially have access to if we apply and get approval. So, and this is a specifically very, very interesting cohort, MOBA, it's in Norwegian, stands for Mother and Child. It's a trios cohort, and to the best of our knowledge is really large, maybe the largest in the world cohort of trios. So we can, and again, we have all the registry data that I mentioned available to us in that cohort. So in total, it's, I think, 280,000 in MOBA and around 240,000 will have the genetics data. So it's especially interested in trios because then we can look at the transmitted and non-transmitted alleles. Because we know the exact genetics code for the parents and their kids, we can look what alleles do they got from parents and how that affects the status of the disorder or other phenotypes. But we can also look at the non-transmitted alleles. These are the genetic material that is not transmitted from the parents to the kids, but it can shape the environment that kids live in, so it can also have this indirect effect. This is very interesting to look at. So beyond MOBA, we also have our deeply phenotyped samples. These are clinical samples, so MOBA is a general population, but deeply phenotyped samples where we have roughly 50-50 proportion between cases and controls is a very good data, and there, in addition, we also have brain imaging data. So it enables very interesting research. Quite recently, like if I recall around 2 years ago, we got access to the UK Biobank data, and this is a really game changer in this field, because in total it's 500,000, 500,000 cohort of generally healthy population between 40 and 45 or 40 and 50 year old, and they have genetics data available on everyone. So this is really impressive, probably the largest single cohort genetic study in the world. And they also now release the whole exome data on 10%, but this 10% is already 150 terabytes on disk, quite hard to work with. And then they also have the goal of doing the brain imaging on 10,000 participants. So right now we are at 43,000 cases in UK Biobank, and it keeps increasing. So, and by brain imaging, I mean all the three modalities with the structural images, diffusion, and resting state, and like task-based MRI. So we also work with ABCD data. This is 12,000 kids from US recruited around 1009, and they will be followed up. It's a longitudinal study, so they will be followed up until they, like 25, I think 25 year old. So also very, very interesting, especially for us, because in psychiatry, many conditions, specifically schizophrenia, is developed in adolescence, so between 15, 17, and up to 30-year-old. So looking at this cohort, seeing who will develop, who will convert to schizophrenia versus not, we can sort of look at this data later and investigate what is going on. So, and they also have the genetics data, so kind of, and all of them have brain imaging data. So really interesting study for us. So our collaborators include the PGC, which is Psychiatric Genomic Consortium, where we contributed our Norwegian samples. It's a very global activity where they collect for all psychiatric conditions and even beyond, like Alzheimer, for example. They collect not necessarily samples, they also work through meta-analysis similar to ENIGMA, which is kind of a worldwide consortium in genetics of the brain. So it's really nice to be part of the PGC, being able to access a network of people that work with them on specific projects. And also we do genotyping with Decode in Iceland. So Decode is an Icelandic commercial company that do genotyping, and yeah, so we work with them. And also San Diego for development of statistical tools with Anders and Dahle. So really good for us to kind of have very good data and work with our collaborators, kind of to get the synergy, these buzzwords, to kind of get us to our aims, which we think are quite important. So of course, and by the way, please ask questions at any time. So if I go too fast, if you want me to sort of elaborate on something, right? So please don't hesitate. I think we can do it quite interactively if you want. So data sensitivity is quite important because probably the genetics data and psychiatric data is the most sensitive data ever. Like I would probably be okay with leaking my bank cards and the passport, but not the psychiatric conditions, right? That is really sensitive if that gets disclosed. And also there is no way to truly anonymize the genetics data because that's like anonymous fingerprint, right? It's the identifier, right? Looking at genetics data, it's totally possible to identify a person. Maybe it's hard to connect it with like the registry, personal identifier, name, etc. But it is a very big concern working with this data. So we store it in a secure cluster in Norway, and even within Nordic countries, we use a special system called Trigve to collaborate. So no individual level data goes across countries, but we can sort of formulate queries and get a specific questions returned to us for further meta-analysis. Yes. So moving a little bit more towards the specific projects developed by my colleagues in the multimodal imaging genetics group. So this is a group. These are my colleagues. We work at quite a few cool projects. I think some of you may know Jarek. He'd been here in MIPT for a seminar like a month ago. So yes, their major aim for the group is to identify factors that harm and protect the brain. And it's definitely relevant to mental disorders because, as I mentioned, they're early onset disorders, and that coincides with major changes in brain. So why is that happening? We probably need to research more in that direction. So the project span across all of the modalities, including structural MRI data. So we normally work with 3 Tesla scans of the structural MRI data, including the thickness, areal expansion, so surface area and surface thickness, and subcortical structures. So normally processing the data with FreeSurfer, extracting the features, and then looking how is it relevant to other clinical conditions that we have on the same sample. We also work with functional MRI, including both tasks and resting state, and also the different diffusion tensor imaging. So yeah, I talked about some of these projects. We also have ping data available and a couple of more functional connectome project. It's a big one. So it's another unglown UK Biobank data. So that's huge spike in the UK Biobank, right? Other, I think ABCD is not here. The slide was probably before. This is our in-house top sample, which is to some extent more interesting than UKB because, as I mentioned, it's a clinical sample. It's 50-50, lots of cases and controls. But UKB is definitely a very, very huge, and it's old, right? I mentioned now it's 2 more batches since we generated this slide. So across different samples that we have available for the imaging data, it spans from, so that's the age, right? The distribution of age in different samples. So that like UKB, you can see, I think this one is UKB. So UKB is specifically around 40, starting with 45 and slightly older. Hunt is our aging cohort where we can study dementia, and the top sample somewhere here in the middle. But basically we have all of the spectrum from 3-year-old, I think, up to almost 98, I think, year old. The breakdown for the different diagnoses would be the schizophrenia. It's really big, with more than 1,000. It's an autism spectrum disorder, Adhd, bipolar, multiple sclerosis. So these are the domains that we're interested in. So the first specific project I would like to mention is by my colleague Tobias Kaufmann, basically asking how old is your brain, right? We can try to predict the age of the person, which is the same as age of his brain, based on the imaging data, neuroimaging data. In this case, it's FreeSurfer extracted features from the raw T1 images across UK Biobank individuals and other samples processed in Movement. So we can build the models that just try to predict the age based on the neuroimaging data. And that's interesting because once we apply this model to a new sample, we will estimate an age that is different. Like you can say it's just a model error, right? But it actually turns out to be quite biologically relevant because, for example, for dementia, the brain age gap is positive. So brain age gap is a difference in years between the brain age predicted by the model as opposite to the real age that the person has sort of recorded. So the positive brain age gap means that the brain looks older than the person's real age. right? And it's quite natural that in dementia, in the cases of dementia, the brain looks older. Yes? Please.
Brain activity depends from age.
So the question was whether brain activities depends on age. And not that's a good question. In this case, we are looking at the structural MRI data, but I think it's also known that structural MRI changes over time, right?
Big changes.
Well, I think it's considerable changes, definitely. So the brain grows during the neurodevelopmental phase, and then it's kind of neurodegenerative stage after 30 or 25-year-old. I don't remember, but and I think they controlled for that. So they looked at they validate. I just remember that they extensively validated the model. I'm sure that validation in terms of like the fact that the training set was collected across different age span was covered here. But it's an important consideration. So also, in addition to using the brain features across the entire brain, they could build the model only using a subset of the features, let's say, for example, in the cerebral cortex or in the frontal region. So kind of to see if the age of the brain in a specific region is further associated with different neurological or psychiatric conditions. Right? Just one idea we can probably extend by not going through the raw T1 images, but directly feeding the raw T1 scans.
Is it correct that we use as features various volumes of various brain parts?
So I think in this case the input features are maybe like roughly a couple of hundred features extracted by Frei surfer. It's cortical, maybe also subcortical structures.
Right. See this surface.
Yes, exactly. But it would be interesting to feed directly the T1 scans and see. So, moving to another project. I think it's again UK Biobank data. So basically, with Frei surfer 6, there is now a feature to sort of estimate the subfields in a hippocampus, right? In total, like I think roughly 13 features. And then so after running this analysis with the new Frei surfer, they just run a very, very standard genome-wide association study, which I will talk about later. And they found 8 novel loci, 15 in total, so replicating 7 of existing findings and looking at the novel loci and kind of describing the novel loci. So, for example, hippocampal tail seems to have quite specific genetic architecture. Like the stars here means genetic locus is associated with whole hippocampus, so its architecture is at least explored from before, but the hippocampal tail, that's a totally new kind of set of consistent findings. And some of the locuses were pleiotropic, so associated with multiple conditions. That was also quite interesting. And the matrix on the right is the correlation, both phenotypic and genetic correlation across these features. So, as I'll just briefly go through the other two projects. It was about involvement of cerebellum in schizophrenia and about the importance of more advanced statistical models where we look beyond the mean. So normally in statistics you would have two features and you associate is one of them correlates with another one, so it's kind of comparing the means and see are they different. But here they've looked at the heterogeneity in schizophrenia group. So it turns out that the brain volumes were significantly more heterogeneous in schizophrenia group. That is in addition to a known fact that on average there is a cortical thinning in schizophrenia, but beyond the general reduction of the cortical thickness, there was also a larger heterogeneity in schizophrenia group. So this is a quite interesting way to look at this data, suggesting that more nonlinear models are important in this context. So I guess now we can move, unless you have questions for the previous part, can maybe move to a more, yes?
Can you please explain what does it mean heterogeneity? What deviations from mean?
Yeah, so this dispersion model that he is studying is, so basically the variance of, so if you're asking what regions, that's a good question. I think again that is a volume, maybe also the surface area and surface thickness, but also volumes of different brain structures. How are they associated with schizophrenia in terms of the mean value, but also?
My question was more about what is the mean brain? If we have this model,
No, in this case, we actually have access to the individual brains, so we didn't average them. It's more like after extracting the specific features, do we see a pattern that basically there was, they called the article was called the dark side of the mean, right? So by just looking at the mean surface area or mean surface volume, we are losing the heterogeneity in schizophrenia group. But I think it's published in JAMA Psychiatry now, so we can look more into that. Yes.
So you told that you have like countertops scans, yeah? So from what scan, unlike just one neurologist or two or more of them to make their decisions, I mean, independent. So is there real changes or not?
So I'm not sure I fully understand the question. Is it about do we have samples with multiple diagnoses for the same person? Okay. I mean, yes, that happens. I did not specifically look myself in our data, but I know that, for example, in the ISAC study in Denmark, this is kind of a major issue that they had to address, because especially in psychiatry, people are classified with one disorder and then sort of 2 years later they change the diagnosis. So people can convert from schizophrenia to bipolar or vice versa. They can also frequently get ADHD. And it's in this case, it's always like, because more than 60% of the people in our center are with medical degree in psychiatry, we talk to them to exactly understand how should we define our cases versus controls. If you study things I mentioned, like treatment-resistant schizophrenia, it's always like very tricky clinical definition. Is it treatment resistant? Is he a non-responder or not? So it's more a clinical question, and we work with clinical people too.
Was it not about psychiatry but neurologists? So was it analyzed by some specialists, not just one, to make it objective?
For neurodegenerative disorders, you mean dementia, for example, right? Or it's. I think it depends on the study, so depending which one do you refer to. If, for example, in UK Biobank, the dementia was actually a proxy to the dementia. It was more like, because in UK Biobank, we only have access to the people mainly up to 50-year-old. There is no dementia in UK Biobank, and they've used a proxy. So basically, did your parents have a dementia? And then genetics of the person, because we know the genetic of a kid will be highly correlated with genetic of their parents. So they've used this kind of connection to establish a proxy. I think it's always like discussable, the exact definition. In our case, I think we have a clinically diagnosed dementia. It's more a question about controls, because in dementia it's very hard to define that the person does not have a dementia. He must be old enough to conclude that he's a good control. So I think it's... So okay, let's move to the second part, which is kind of no research here. It's more a general introduction to genetics of the complex traits. So we will go through the very basics, like what's the difference between Mendelian and polygenic inheritance, because complex traits in this context mean that they are influenced by multiple genetic variants, and Mendelian is single monogenic disorders. So I'll explain the terms, and we will look in how people are doing it right now with genome-wide association studies, and sort of also discuss the concept of heritability and how it gets estimated from the twin studies. So, first a small disclaimer. So, I know many people are involved with bioinformatics, and I would like to kind of draw the distinctions that somewhat these two terms are not the same, because I heard this confusion. So genetics is a branch of biology concerned with studies of genes, genetic variation, and heritability, and the term is coined by Mendel in like 1080s, 1800s, studying some peas, flowers, etc. And the bioinformatics is a lot more recent field, probably 50 years or so. All that is, like from the name, it's bioinformatics. It's interdisciplinary field that developed methods and software for understanding biological data. Sort of in the context of genetics, this term has its own meaning. It refers to the pipelines that we use in genetics in order to process our specific data. For example, like the candidate gene analysis. So basically, these are quite related. In genetics, we use a lot bioinformatics, but like not quite the same. And this is just a general example of what I'm talking about. The details are not so important, but this would be a typical genetics study from the PGC consortium, where they thought to find the differences between schizophrenia, bipolar, and healthy controls using the bioinformatic tools and just using purely genetics data to cluster them into clinically relevant subgroups. So we'll probably talk about it later. So moving to the genetics, I think you've heard that we as humans have many genes. It's around 24,000 genes in humans, and they exist in different forms depending on which form of a gene, so which allele we have. It defines like eye color or hair color, etc. So now we actually have access to the technology that can genotype or sequence people. And it's an important distinction, both conceptually and in terms of the cost. So sequencing or deep sequencing means that we'll actually read the entire human code, the entire person's DNA, which consists of 3 billion base pairs, right? So this is a book of these ACTG letters. But a lot of the code is the same across people. So take everyone in a room, we will share the 99.9 something % of the DNA material. So in terms of genetics, where we study variation across people, it's kind of more interesting to look at the specific locations in the DNA where potentially we might face a different letter, right? And such letters called single nucleotide polymorphism, where just one letter can differ. Everything else is the same, but in this particular location in the chromosome number, let's say 10, there is a difference, right? And there are specific technologies for genotyping. Using Affymetrix or Illumina chips, we can actually pretty much quite cheaply do the genotyping. So that's the data we mainly work with. So not the deep sequencing data, but the genotyping data. And speaking of Mendelian versus complex traits, so this is an example of disease called cystic fibrosis, which is a Mendelian autosomal recessive inheritance. That means that if we look here at this mother and father who are carriers, they will have one allele that is broken, so one copy of the gene is broken and another gene is healthy. And there are 4 potential outcomes for their children. Either it will be a completely healthy children, unaffected child that has 2 copies of a healthy gene. Or it could be a carrier gene. There are 2 because he can get a broken copy of a gene either from the father or from the mother, right? But they will not exhibit any symptoms. So they will be carriers, however, they will be apparently healthy. And it's the 4th case when the child receives 2 broken copies of the gene, then he will receive the disorder. So in this case, we know the exact location of the gene. It's this CFTR and chromosome number 7, right? So this is a simple known pattern of inheritance for this disorder. In some cases, it's a bit more complex. For example, it's just one copy of a gene that is sufficient to get the disorder, like in case of Huntington. But in some other cases, you actually, like one copy saves the person. He just needs at least one copy of the working gene, and then he doesn't get the disorder. But the most traits that we are looking at look like that. So instead of one specific gene, like it seems that there are genetic factors that are relevant to the disorder all over the place. And we call this polygenic inheritance. Polygenic means multiple genes involved, right? So, and they have very complex patterns of inheritance because sort of, these chromosomes, they cross and they can be inherited from mother or the father. So, like, there are it's not a discrete number of outcomes that we are looking at. And height is a very good example. So you can, of course, argue that in schizophrenia, the diagnosis is a little bit kind of unclear. It depends on the person who is giving the diagnosis. Height is extremely easy to measure, right? You have the ruler, you measure how tall is the person, and that's it. And yet height is also very polygenic. It's many, many genes involved in the etiology of height. According to our estimates, maybe just 3 times less than in schizophrenia, but still a lot. And it's also a complex interplay of environmental and genetics factors. So environment plays a very big role in these disorders. And yes, so the majority of traits in our domains are more like this, right? So really complex to study. So, but now, because we have access to the raw genotyping technologies, we can establish a very large cohorts and try to find the association between traits. So, for example, in this case, we are looking at the case control study when half of the samples are cases, meaning that they have the disorder, let's say schizophrenia, and the other half are controls, right? Here, like above the line, controls. And looking at the specific variant G1, we can probably conclude that, you see, in controls, they mainly have G, right? So G seems to be a healthy copy of the gene, but cases, they're 50-50, right? So probably having the A letter, it's kind of, it could shift your risk towards having the disorder. So, and for example, for this other locus, when we have roughly 50-50 in both cases, so it doesn't seem to be associated with the disorder. So that's what we are looking at in genome-wide association studies. We look at the different, for each specific variant, we count the genotype in the healthy group and in the group affected by this disorder, and we are looking for statistically significant difference, for example, with the exact Fisher test or any other methodology.
But you do this for each site separately, right?
Absolutely.
But then you apply correction, of course, right?
Of course. The multiple correction is, in this case, the standard p-value would be 5 times to the minus 10 to the minus 8.
But what kind of correction? What kind of multiple hypothesis do you use? Is it just Bonferroni correction?
Bonferroni.
But there is some other approach to rejection based on average rejection rate. Why don't you use that?
So one particular reason is the field is quite conservative about that. So like the GWAS is a large game that many consortia are playing, and then with this estimated number that there are 1 million independent sites in the genome, and therefore they just divided 5% from Dr. Fisher by 1 million, and they get the 5 to the minus 8. Everyone is happy. But of course, like in our group, we have a method that we use extensively using Benjamin Holder FDR, kind of more inspired by Efron FDR, not Benjamin Holder, sorry, Efron empirical FDR. And it's discussable what kind of multiple correction testing we could get to reach a better statistical power. However, the very classical GWAS, it's like the rules of this game is 5 times to the minus 8. I totally understand that there are better ways of doing it. But as long as it helps people collecting samples, I think it's really good that people establish, okay, this is kind of, it is conservative, but there is nothing wrong with it, right? It works. So, especially if they discover, at first stages, they discover maybe 1 or 2 variants when it's a growing GWAS and a new phenotype, and then it's not that bad, I think. But we'll come back to it, right? So here you see the dashed line that is about discoveries. This is a decimal logarithm of p-value. So around 7.3 is the genome-wide significance threshold. And I just wanted to highlight it here, how different is genetic architectures between schizophrenia and the putamen volume. So this is the GWAS from ENIGMA, released in 2016, I think. You see, the most strong p-value is approximately the same. It's like, okay, if you disregard MHC hit, it still will be like 10 to the minus 20. But here it's just how many? 5, right? 5 loci discovered in ENIGMA versus 108 loci discovered in schizophrenia. So this is an example of different polygenic architecture. Schizophrenia seems to be more polygenic. There's more hits discovered in this case. So our model, the way we believe it works, is this simple additive genetic model, right? So if you think of Y as a phenotype, let's say height, right, in centimeters, we believe that every locus in the genome have certain contribution beta, and depending on how many reference alleles do we have for that locus, it will contribute to your height. Or in this case, I'm just making up this example of how tall is the head for the smooth, right? So the smallest one is 6, and that if you have all the capital M and N alleles, if he has lowercase, if he has a capital alleles, each contributing plus 2 centimeters, then it's a tall head. I mean, head should be biological, right? It's not quite clear. And the intermediate would be 8, right? So just an additive model, depending of what are the alleles at each locus, you just sum them up. And we can, so once again, Y is height, G is genotype, the number of reference alleles for a given locus, and the beta is the genetic effect, right? Once we can learn the genetic effects from the GWAS, we can turn that into the polygenic prediction, the very, very basic linear models that will predict predict the height based on our knowledge of genetics. And we will talk later about this model, how good is it at this current stage.
So, by saying, so GWAS, it's what? It's from the previous test, right?
Betas.
Huh?
Betas or, sorry? GWAS. So, yes, so G is Genome-wide Association Study. It's this whole concept where we will test marginally each SNP by itself, each genetic variant we test in isolation versus a phenotype, basically estimating the Pearson correlation. So we can use the genetic effects from the Genome-wide Association Study to build our predictor model. And like so far I've been talking about the quantitative traits like height, right? So we can try to predict the genetics of height, but it also applies to the disorders under this liability threshold model. So we believe that the genetic predisposition to a trait is a continuous, it's a spectrum, right? How many or what is the total accumulated risk of having schizophrenia based on genetics? And once the risk or liability exceeds certain threshold, then the person converts to actually having the disorder. So this is called liability threshold model, and under this model we can apply the same methodology to study the genetics of schizophrenia. So in this case, it's also quite important to take into account the effects of environment. So in case of Mendelian disorders, it's mainly like the gene decides. If there are two broken copies of the gene, then the person will get the cystic fibrosis. In this case, the role of environment is also important. So if we, from our previous prediction model, sort of thought that the smooth head will be exactly 10 centimeters or 6, in reality, it will be kind of a histogram around 10 plus minus, right? Because depending on the nutrition and the other environmental factors, there will be some noise around this genetically determined height. And same with other cases. So it's a very important question, how do we estimate the proportion of genetics that is due to environment versus the proportion of the phenotype coming through environment versus coming through genetics. In other words, like we're really interested how heritable is schizophrenia, right? So one way to address this question would be to look at the twin studies, because Mother Nature set up a very beautiful experiment for us. It's twins, right? Twins could be identical, or they also identical monozygotic twins versus non-identical twins, dizygotic, so born at the same time, but as different or similar as normal siblings. And so the way to use this data for studying heritability is somewhat subtle. We need to look for the differences between monozygotic and dizygotic twins, because if there is no genetic effect, then mono and dizygotic twins will be pretty much similar to each other. But if there is a strong effect of genetics, then because I didn't mention that identical twins share the entire 100% of the DNA material, versus the other twins, they share only 50%, like between brothers and sisters. So if there is a difference between, let's say, height or schizophrenia risk between mono and dizygotic twins, it has to be due to genetics. Everything else should be pretty much similar. They lived in the same family, they shared environment, and if there is a difference, that one should be due to genetics. And it's, for example, a pretty famous example is schizophrenia, Genian quadruples. It's 4 monozygotic identical twins born like 90 years ago, and they all developed schizophrenia. And current studies suggest that the concordance rate between twins are highly different. If it's a dizygotic twins, so like ordinary siblings, then it's only about 4%. So if one has a brother or sister affected by schizophrenia, it will be around 4% chance that the second twin will also develop the disorder. But in monozygotic twins, it's much higher. So for us, it's a very convincing evidence that there is a very, very large heritability in schizophrenia. In terms of heritability, like variance explained by genetics is around 80 to 90%. And speaking more about, because basically it's an entry point. If a trait is not heritable, then there is no interest for us to go in genotype, invest in genotyping and do the studies. But almost everything in psychiatry is highly heritable. And the height is not in psychiatry, but it's also like as heritable with narrow sense twin heritability of 80%, right? So this is according to the twin studies estimates, the amount of additive genetic factors that they explain in heritability is roughly 80%. And then we start kind of losing the predictive power if we, from the twin studies, go down to, let's say, GWAS data, right? If we try to estimate heritability from the underlying genetics data that we have from genotyping, then we will get a much lower estimate. So this red bar here indicates missing heritability. So we know from studying twins. So twin studies, they were around like 50, 60 years ago, because you actually don't need to genotype people. It's an observational study, and you just see what is the like prevalence of the disorder between twins. But now we have genotyped them, and we can't actually explain more than 20%, like 20%, right? This big gap that we observe across many disorders is disturbing, right? We know it's heritable, but we know exactly how is it heritable. Further down, once we start predicting using the genetics data to do prediction, it's even lower, right? So that blue bars is how much we can sort of bring back to clinic. If we can, from the genetics data, do an estimate that is like 80% accurate, we can actually go to the clinic and start doing the stratification models or prediction models, but with 10% it's still not clinically relevant. So I think we could have a break after this slide. So let's just briefly look at what are the ways to estimate the beta, and it's only heavy technical slide with formulas here. But so as I mentioned, additive model is where the phenotype is just a linear combination of the genes that a person has, right? And we in GWAS are interested in beta. And so the real challenge here is that this genetics matrix is huge, right? So in the second half of this presentation, we will look at different statistical methods, but the underlying mathematics is really simple. We are talking about the linear model, where the phenotype is linearly related to the genotype matrix G that we know about, and the unknown beta effects that we don't know anything about. So the typical dimension is the number of individuals now approaches 1 million. For height, it exceeds 1 million. For schizophrenia, it's 100,000. That's N. For the N. For the number of genetic variants, it's 10 million. So we typically look at, if you include all common variations of variants that are... 1 million, let's say. 1 million. 500,000 UKB, 1 million pooled together across different sub-studies, or specific problems like psychiatry and major depression, clinical samples. Because of the PGC and other huge consortia, it's up to 100,000. I think depression is 300,000.
Okay, so you will talk further about statistical approach.
Yes, that's the second half. It's sort of to give you something to think about during the break, and then we'll continue with kind of a bit more involved, heavy mathematical part.
This was a break, Oleksandr, a fantastic lecture. We enjoyed it. But for the audience, will you use the notion of penetrance and epistasis for your analysis, or you just ignore it?
No, we do ignore, quite unfortunately. I'm aware of that. That would be extremely interested, but I'm not aware of the successful studies that have proven that, like, okay, we now observe and study the epistasis effect. If you can point me to the studies, I would be very interested to read through. And I know it is implemented in some tools like Blink. So so epistasis and gene-environment interaction is something that people are generally very interested in. But I think it's still maybe in a larger sample size, or maybe specifically in psychiatry, it's difficult to observe because it's so complex and polygenic.
But in penetrance, if you analyze twin studies, then the user can interpret it as a different environmental effect, or as a stochastic different penetrance of the effect. So I think your models will benefit a lot if there is a way to include this variety.
True. I think the models so far mainly like focus on the clinical samples, trying to understand what we observe in exactly the clinical samples we have.
So for the audience, everybody understand what penetrance is? Yeah? So we have a break.
Yes. Коллеги, давайте продолжим. Да, мы остановились вот на этой математической задаче, и теперь переходим к более трудоемкой части про статистический анализ So challenges and advances in statistical analysis of genetics data, hopefully with applications to the brain imaging data. So yes, we will talk about the burden of multiple testing and how do we address it with multivariate analysis called MOST test. And we'll discuss a little bit about the cross-trait genetic architectures and sort of with two tools we recently developed, the mixer and the conditional FDR analysis, and then a separate conclusion section on the polygenic risk scoring. So yes, we've discussed that most traits are polygenic, so there is not a single gene involved in the etiology of the disorder, but rather a whole spectrum of genes across all over the places, right? And we do the genome-wide association studies, testing each variant in isolation. Speaking of the burden of multiple testing in this case, I mentioned that in the standard genome-wide association study, we test around 10 million of markers, genetic variants in isolation, leading to around 10 million of the genetic tests. They're not all independent, but still, it's really lots of hypothesis testing that is going on. So speaking of multiple hypothesis testing, I mean, this is really basics in statistics, but we talk about p-values, right? And generally the meaning of p-value is how unlikely is to observe a given event by chance, right? So p-value of 5%, which is a commonly applied threshold, is that this probability of 5% this data is observed by chance. But if we repeat the same test 20 times, right, then just by luck you would expect that on average at least one p-value will be below 5%. So this is what these two histograms represent. If you repeat, so this is a histogram of the p-value if you have repeated them many times, right? And on average they will be uniformly distributed. So you do expect that just by luck out of many tests that you've conducted, some will be low, right? So this horizontal bar is a p-value, and you see there is like 500 of really low p-values here. So in order to, once you do multiple testing, it's really important to observe kind of this skewed distribution. If you see that there is a kind of a heavy tail, a lot of small p-values, more than uniform, then you can conclude that you are observing an event. And in our domain, we typically represent this as a quantile plots. So this is a QQ plot from the PGC schizophrenia GWAS. So QQ plot following the null line would kind of correspond, to this case when it's uniform, right? And once the QQ plot deviates upwards, then it means that so you see this one is a logarithm, decimal logarithm minus. So you will observe really excessively small p-values, more than you would expect by chance. So this is kind of the type of data I will show quite a few times, and it's important that when we observe the genetic effect in the whole genome association study, you would see the stronger deviation to the left in the QQ plot. And that was kind of my retrospective on the statistical power. We have approximately 1 million of independent statistical tests, and in addition, we're also working with very small effect sizes, because if you split the heritability into so many loci, like in this figure, right, you can imagine that individual effects are pretty much small, right? So that's why to detect these small genetic effects, small betas, you need a really large sample size in order to exceed this stringent p-value threshold. And there are a bunch of other challenges in working and interpreting the GWAS data. So that's a scale of the data is a technical challenge, and the correlation structure is another challenge. So to kind of the one very straightforward approach to get some robust findings is to increase sample size, and that's the approach taken by many consortia worldwide, including PGC, ENIGMA. We work a lot with 1.1 million GWAS on the education attainment, for example, or the biobank. They're all about growing the sample size to have enough power to detect small changes, even though we have small effects and the Bonferroni stringent correction. So we've developed a tool that kind of helps us to predict the future of the GWAS once the sample size is growing further. So in this case, the axis here in the logarithm scale, like 1,000 sample size, 1 million sample size, 10 million sample size, and the vertical axis is how much of the genetic signal we will explain by the hits that pass the significance threshold, right? So it turns out that different traits are kind of on a very different curve here. So, for example, in order to do discoveries in genetics of the lipids, so this would be low-density lipids, total cholesterol, HDL, and triglycerides, you don't need as much of a sample size compared to psychiatric traits. So, and it's not just because it's more heritable. Actually, I think the heritability of the levels of the lipids is not that high. It's just because it's less polygenic. So these traits are closer to the Mendelian traits. There are not as many genetic variants that define the levels of lipids compared to, let's say, this one, which is Adhd, so the major depression disorder. And naturally, if you have less genetic variants with non-zero effects, then individual variant has a larger effect and it gets easier to discover. So this model kind of helps us to, by fitting the model to the data, we can understand these hidden parameters that we don't immediately read from the GWAS data, which are about the polygenicity and discoverability of these traits. So polygenicity is how many genetic variants are involved in the disorder versus discoverability, how large is the effect size. And so, for example, we predict that so I think some of these curves are schizophrenia and bipolar are almost on the same path here. So we would need almost 1 million cases in order to explain half of the genetic, right? So this kind of power analysis is very important for the field. It's doable, right? It's just even 10 times larger international effort is required to get us there. But there is another way. In addition to collecting more and more samples, we can apply better statistical tools that, for example, benefit from the multivariate analysis. So this slide is like a screenshot of our paper on bioRxiv by my colleague Dennis van der Meer. And we've looked at the genetics of the regional brain morphology. So the run-free surfer extracted the features for cortical area, cortical thickness, and subcortical volume, right? These are the 3 domains. And also we put them all together in one big analysis. And the idea is that the genetic signals that we observe is highly distributed across the brain. So you see, if we work with each ROI independently, and in total here there are 171 ROI regions. So if we process them one by one, let's say, the volume of putamen or some fifth ventricle by itself, right? Then we have yet another dimension to the multiple correction problem, right? In addition to testing 10 million genetic variants, we also test 171 ROI measures. So the multiple correction, at least in terms of Burfironi, will be just terrible. Right, but if we put all of this in a multivariate setting and analyze jointly, just looking not for genes affecting a specific measure like fifth ventricle, but rather looking at the genes that have some effect on the overall brain morphology, then we are back to the same burden of multiple testings that we observe in univariate GWAS. So, and instead of sort of struggling with too many tests, we actually get a benefit from once the genetic signal is shared across brain regions, in this case, the cortical area and thickness, then we can get a boost. And so this was an explanation, but the net effect is pretty amazing. We get 3 times more loci compared to the standard approach using thickness, and in the total, looking across the brain, across all of the 3 modalities, we get from 150 up to 350 loci. And this is like the unit of discovery in this field. Everyone is comparing in terms of how many loci, LD-independent loci people discovered. Another interesting thing is we looked how, so just looking at this green, blue, and red domains, how the genetic loci are overlapping with each other. And together, I think there are around 300 in here. So by putting together these 3 modalities in one analysis, we get even further boost by extra 50 hits, 50 regions together. So boosting the power of discoveries using the multivariate approach is something that people know around for 10 years. Like it's possible to use tools developed in 2008, but they were pretty slow and for some reason not popular. So our main message with this paper was just please use it. It's really handy. No matter what multivariate approach you do in genetics, once you have multiple measures like in imaging, it really makes sense to apply. And same would apply for just vertex-wise GWAS, in this case, vertex-wise imaging measures. So here we aggregated them into regions of interest, but vertex-wise GWAS is also quite interesting. And sort of moving to the third way of boosting the power. So the first one was brute force, collect more power. Right? The second one, this one was to find multiple phenotypes, multiple measures that make sense to put in one analysis and benefit from the distributed nature of the signal like, across the brain. And the third one would be to redefine the ROI regions so they're more genetically discoverable. So I think this is just a reflection on what I mentioned. The more SNPs is there associated with the disorder, the less would be the effect size for a given SNP, right? So the gradient sort of left bottom to right up would be the heritability, right? It's the color. And then, of course, I mentioned that before, if you split the signal into many monocy, individual SNP will have a lower effect, right? So there are no surprises here. But we found that basically this figure partially explains why in the recent paper, the ENIGMA cortical discovered less loci for cortical thickness. That's because, like heritability-wise, they are not that much different. But in terms of polygenicity, there are many more genetic variants associated with regional thickness. And therefore, the effect size, which we call discoverability, is lower. Right? So individual effect is lower. Given the same sample size, it would be more difficult to discover. And now, of course, in imaging, there are different porcellation schemes, right? So in our works, we typically use from the free surface default Desikan-Kiliani atlas with 34 regions. So it was a cortical area and thickness. But there are other atlases, for example, the Chen's atlas based on, so specifically informed by genetics information. And that atlas, I think, has, instead of 34 regions, it has less, maybe 12. But so we also compared with Yao Atlas and the Glasser Atlas, which I think you guys are more experts in these topics, definitely. But my understanding is that Glasser Atlas just has more regions informed by different properties. So what we found here was that the discoverability parameter that we're interested in, right? The larger discoverability, the easier it is to conduct genetic studies on those phenotypes. So discoverability kind of changes. For example, it was the largest in Chen area atlas. Of course, it's kind of an expected result because the Chen atlas was informed by genetics. On the other hand, it's kind of a nice reflection on the fact that depending how do you define regions, you might get different picture in terms of understanding the genetics. And the Glasser atlas didn't perform so well because it was we definitely need to include the analysis of the size of the region, right? Because smaller regions, they're more noisy, more difficult to sort of extract from the imaging data, therefore they're more noisy, and it's quite natural that it's maybe harder to do genetics on those. But at the same time, we've noticed that it's like polygenicity stays the same, so discoverability changes across atlases and heritability changes. So anyway, the idea is we need to look more into the way we do the porcellations of the different brain regions, and specifically once we target the genetics, it may be that some of the previous atlases like Desikan-Kiliani or the Chen's area atlas would be still reasonable to use. Yes, so now maybe we can take a question or two on this topic if you want. Now, so there was some imaging related, neuroimaging related topics. Anyway, maybe we can, after we are done, we can come back to this because I think it's really, really interesting how we do genetics. So moving more towards the kind of changing the topics. Here we looked at the polygenic overlap across traits. So it's a recent model that we developed for the cross-trait genetic analysis, and currently you can stop there. This is just how we fit the model, right? It's nothing super fancy, just fitting the model to the data and then observing the parameters that we represent as these Venn diagrams of unique and shared trait component. So the story here is that normally people, nowadays people do the cross-trait genetic analysis, it's all about genetic correlation. So this is a famous paper in our domain that introduced or enable people to quantify genetic correlation from the GWAS data, and they looked at a whole bunch of different disorders, and they found that like there are these clusters of highly genetically correlated disorders. So quite naturally, schizophrenia, bipolar, and depression form this cluster on the right, but schizophrenia did not correlate with the years of education, for example. So the genetical predisposition of a person to go to school seems to have nothing to do with his schizophrenia case control status. But we thought like both are brain, right? So there's probably something in the brain that forces people to go into, I don't know, master, PhD studies, and also the schizophrenia is also brain. So we looked with using these QQ plots that I mentioned before, and we found kind of an enrichment signal. So once we constrain the list of variants plotted here, we see this leftward deflection, and that indicates some relationship between traits. And even like without talking about QQ plots, we actually find shared genetic regions that are associated both with schizophrenia and with education attainment. So we would like to revise this kind of empty areas on this atlas of genetic correlations, trying to understand what does it mean that traits are not genetically correlated. And this, I think, is pretty nice illustration to this concept. So, for example, like this it's a 2010 pretty old study looking at the incidence rate in terms of like how many per thousand people have A would be schizophrenia and B would be bipolar. So depending, so as a function of their performance at school, right? So the better kids study at school, the lower is the chance that they will develop schizophrenia, right? So here there is a very nice negative trend on the right. But this bipolar, it's on both ends. It's both poor and good performance at school that is associated with an increased incidence rate of bipolar. So this kind of interesting nonlinear relationship reveals itself as zero genetic correlation, because whatever is about this kind of branch and the other branch, it cancels out. So I mean, it's not intuitive, but you see this is a highly nonlinear model, right, more like parabola. And to model this nonlinear relationship, we need another statistical models, not just a simple correlate or not correlate. So, and that was we saw after we developed this model and applied it to the wide range of psychiatric conditions, that yes, schizophrenia and bipolar have a known relation from before, they are highly correlated, we reproduce the same results, but for example, schizophrenia and education attainment, which should be somewhere here, they don't correlate, the genetic correlation is zero, but nevertheless they share some core set of genetic variants that have non-zero effect in both traits, right? So kind of building these models of genetic architecture based on the data that is already available to us is a good way to enhance our understanding of this data. Particularly, it was quite interesting to look at the overlap between schizophrenia and depression. So they are also positively correlated with around 35%. So the R square will be just 10%, but still this is a no significant correlation. However, in terms of like, what genetic variants they share, it's a lot more than we previously expected, I think. So it's nice to explore this further. And it's also the case for the negatively correlated traits like schizophrenia and education attainment. ADHD is education attainment and ADHD, which is attention deficit hyperactivity disorder. So this is another prominent case, both with negative correlation and high studies. I think we can more or less skip this part. It was more the details about how this model works. We've tested it in simulations. It does work nice on synthetic data, and we have a whole bunch of figures from this paper to look in more detail. What's relevant probably in this context is that applying this in the neuroimaging domain, we can see that the total area and the mean thickness across the brain it's not that much genetically correlated. Again, RG of minus 37.37 means that the R square is just around 10%, so there is not much shared variance explained by this. However, in terms of the polygenic overlap that we've developed in this cross-trait mixer, it's really quite substantial. So that was interesting. Another tool that we use, and by the way, the cross-trait mixer and this tool that I will speak about is publicly available. It's on GitHub. It's on the GPLv3 license, so it's free to use, and it's applicable to the publicly available data. So speaking of the genetic overlap, we also developed the tool maybe 5-6 years back to use a conjunctional false discovery rate inspired by Brad Efron empirical false discovery rate, empirical bias. So applying this technique in our domain, we had a series of publications with different like schizophrenia versus bipolar, and something in neurological traits and also the cognitive domains. So really very smart, productive technique to look at the traits shared between different disorders. So yes, and the idea is maybe better understanding of the cross-trait relationship can help us to point us to maybe some drug targets that could be further explored, etc., and eventually improve the polygenic prediction. So I think it's quite quiet audience at this point. Maybe now it's a good news. We are moving towards the last topic, which is about polygenic risk scoring. Looking at the Genome-wide Association study for schizophrenia, in 2014 they published a major paper discovering 108 loci. Now 5 years later there is a paper in preparation discovering 256 loci. So we didn't know what 108 do exactly. Now we have a lot more. So how do we deal with that? How do we actually benefit or try to bring that back to clinic, right? And the concept is this polygenic risk score that I mentioned before. The name is really not a very good name because we can also apply it for, it's best to call it just a polygenic score because same technique we can apply for prediction of the height, and there is no really like a risk of being tall, right? So this is just a polygenic prediction. As I mentioned, we just take the genetic effects from so this odds ratio from genome-wide association study and use it to come up with a single score, so one number per individual that tells the genetic predisposition to schizophrenia or like the height in centimeters. So the odds ratio here, I can remind just odds ratio in a given cohort. If you talk about the odds, right? What is the genetic, what's the odds of having schizophrenia? If you have a given clinical sample, you have people affected, and you divide the number of affected people by the number of unaffected. That's odds of having schizophrenia. Now if we have two groups, we can, like let's say a group of the high risk and the group of low risk. We can look at the ratio between odds. How much more likely is to get schizophrenia in the group that is more at high genetic risk versus low genetic risk. And we can see this again from schizophrenia papers that the odds ratio of having schizophrenia kind of increases with the decile of the polygenic risk. So sometimes it's up to like 20, right? So if you compare the individuals at the highest 10% risk versus individuals at the lowest 10% risk, then the odds ratio between those will be around 15. So maybe 15 times higher risk to get schizophrenia in this age of spectrum. And because this seems to be a very promising technique, the number of citations really skyrocketed. So this is just a number of papers citing the polygenic score or polygenic risk score. And like the publication in Psychiatry by PGC was somewhere in this 2010, I think, where they introduced the polygenic variation contributes to the risk of schizophrenia and bipolar. And since then, it just really skyrocketed up. So the idea behind the polygenic risk score, regardless of like simplicity of its mathematical methodology, is that so maybe we know already a lot from clinics, right? We know that, for example, taking the coronary artery disease as an example, there is a lot of clinical variables that we can use for the risk prediction. But sometimes the risk would be kind of very, very just below the action threshold. So just below the threshold where you could invite people for clinic to do additional follow-ups, additional studies. But if in addition we can provide at least some prediction using the genetics data, we can further stratify people into those having clinical risk and low polygenic risk, intermediate and high polygenic risk. And suddenly having this information will exceed the action threshold and you can invite them for further studies. And sort of it works reasonably well in some other domains, for example, breast cancer. So now you see the same curve of the odds ratio really depends on the quantile of the polygenic risk. So the lowest 1% is significantly below 1, and the highest one is kind of approaching 5 times higher risk, I guess. So they even started a clinical trial, clinical trial specific, just using the polygenic risk score to see if people can benefit from sort of knowing the genetic predisposition to the cancer. And another domain when it looks successful is the Alzheimer's disorder, so dementia. So basically here the area under curve is around 75%, and that's considered to be like a threshold for maybe starting some clinical assessment. We've developed our own complementary tools, which we call polygenic hazard scores, and they are based on the same concept but designed for time to event. So, for example, in Alzheimer, it's not so much important whether the person gets it or not, but rather what age. Because I think if you live long enough, up to 120-year-old, maybe everyone gets it. But here, it's just using the genetic information, we could stratify the Alzheimer sample into like low risk, the top 2% lowest 2% risk get on average 20 years later the diagnosis compared to the group that is lowest risk, right? So that was really interesting to see the stratification in this sample. This was sort of in a very clinical sample, but we see it generalizes in UK Biobank. Again, as I mentioned, there is no dementia in UK Biobank, but we can use some proxy to identify based on parents. And same stratification, kind of depending on the PHS percentile, we see a large stratification. Sorry, that was prostate cancer. I'm sorry for that. So these first two were about the prostate cancer. And we repeated that in Alzheimer, so that's quite natural. If that works in the clinical, if that works for the normal polygenic risk, it should also work for the polygenic hazard score, which is time to event. And it also works if we include more and more data. So now I'm kind of moving towards my final message. It's really good to include more and more data in this analysis, because then we get a higher stratification and better prediction. So that was in other domains like neurology and cancer. So what about psychiatry? This plot shows the performance of the polygenic risk across different PGC studies. Basically the first one in 2009, explaining up to 3% of the risk of the schizophrenia variants based on purely genetics information, right? So, and this is leave-one-out study, so kind of an internal leave-one-out cross-validation study. So here they don't use the same data for testing and prediction. It's a fairly estimated number. Once they grow to the PGC wave 1 around 2010, I think they end up with roughly 10% accuracy of the prediction, and then it's in the 2014 it's grown up to 20%. So that looks quite promising. Now the upcoming release preliminary data is that it's kind of reached a plateau, but there is an important improvement here on the left. But anyway, once we start looking into another clinical setting, the predictive performance goes a little bit down to 10%, and the message is that basically vast improvements are still required before this can become clinically relevant. Even though it is strongly associated, the genetic risk is strongly associated with schizophrenia diagnosis, it is still not close to suggesting to apply it in the clinic. And so these papers that I mentioned from before, it's doing kind of interesting analysis with schizophrenia risk and with bipolar. So schizophrenia risk here on the vertical axis and the bipolar risk here in the bottom. So even though we don't have large GWASs on some of the clinically relevant symptoms, like maniac symptoms in schizophrenia or negative symptoms in schizophrenia, which I've talked before, and the psychosis in bipolar disorder, we can use existing polygenic risk scores of just the overall diagnosis, schizophrenia and bipolar, to predict these kind of symptoms. And sort of this makes sense, this observation that the effect size of bipolar polygenic risk is associated with maniac symptoms in schizophrenia, because bipolar disorder is this mood changing disorder where sort of a person goes through the depressive stage and the very active stage of the mania. So, and the maniac symptoms in schizophrenia seems to be associated with an increased risk of bipolar disorder, while psychosis is sort of associated with both. So using genetics data seems to provide some link to the relevant clinical questions, but it's still very early to do a translational study and put it into the clinics. One particularly interesting topic to study with genetics risk would be the treatment resistance. If I can predict that people will not respond to the treatment early on using genetics data, so that we can maybe shorten the time that they spend on their trying very, very different drugs before they land, let's say, on clozapine. So clozapine is a very heavy drug for treating schizophrenia, and I think it's not prescribed until they've tried a lot of other drugs and see that they didn't work. So if we know from genetics that other drugs will not work, we can maybe suggest to apply clozapine earlier and save them someone from hopelessly trying a couple of other drugs. So treatment resistance is one area where particularly applying polygenic risk score could be like one of the next directions. But that was all based on a very, very simplistic model where we just sum the effects of risk alleles in a linear model, kind of naive Bayesian approach. So if we take but the data is really suitable for any machine learning application where the class, in this case 0, is a case, so schizophrenia, for example, and 0 is control, needs to be predicted based on many SNPs, right, in 10 million SNPs. And we are not even looking at the second order kind of relationship between SNPs, just look at one by one in this case. So just my own speculations on the topic, why basically we convert to statistical modeling, because our data seems to have really low signal-to-noise ratio, which is good for statistical modeling, but maybe not the perfect scenario for the machine learning. So, and machine learning typically require even larger sample size. So even here in our simple statistical analysis, the small sample size is in the order of millions. So maybe once we collect billions, we can go to the classical machine learning. But I think it starts shifting sort of towards that direction, maybe approaching the amount of data that would help us to apply the machine learning, more complex model, deep learning, in order to get better prediction instead of highly manually curated statistical models that we use today in genetics. So yeah, we do some coding in Python and C++, but also many people use R and the classical inventory of the statistical tools. And you're expert on the left here, right, with many techniques developed for modern machine learning. So maybe if we try to apply those more in our domain, then we can sort of address this problem better. And this is like the big data pipeline that basically I think the future is to connect all of these different modalities. Is genetics data, is clinical variables collected from, for example, mobile applications and neuroimaging data, EEG data, polygenic scores into one big model that will therefore get a better prediction at the end, right? So with that, I would like to thank my colleagues who really are supportive in working on these projects. So both colleagues in Oslo and colleagues at UCSD, research fundings, and it's always very important for us to work with study participants who basically enable this whole research by contributing their data. So really thanks everyone. And with that, thank you for your attention. So do we have time for some questions?
One moment. We need to use a second microphone to proper video recording from the questions. So if anyone should want to ask something, you could do it right away. I'll pass the mic.
Thank you for running this lecture with plenty of interesting examples. Could you please open one of the last pages with graphs this progress?
Please stop me.
This one. The previous one. No, the previous one. Yeah, this one. Yeah. So, what's your feeling? What's the key ingredient for this steady, more or less steady progress? The amount of data available, or some kind of more advanced pre-processing, extracting features, some new features, or some new statistical approaches?
Yeah.
Maybe something else.
I think in this case it was the increase of the sample size. So PGC collected from here, I think it was like 3,000 or 7,000 cases, 34,000 cases, and then we get up to 70,000 or 80,000 cases. So I think it's the first step before we can start even thinking of better statistical models, is to collect enough data so we have some flexibility in terms of how we analyze it. So more samples is the first step. But now we are in the stage when better statistical models, as I showed this, for example, MOSE test is really the way to enhance and get to a new level with these current sample sizes. Because doing statistical models is pretty cheap, right? It's one person and a cluster, like you have access for a million CPU hours, you're done, right? Collecting samples is really expensive, so it's the future is to all do both, I think.
What's your opinion related to some more tricky phenotypic features which can be extracted from, for example, structural MRI scans, like some non-default morphometry features?
So we've seen with, for example, with this multivariate test, we've seen that basically by putting together up to 100 or 170 features in one analysis, we really get threefold boost in terms of the loci discovered. We need to translate it into this improvement in terms of predictive performance, but we saw that the more features we put together, if they're perfectly correlated, then it doesn't really help. But once they sort of measure a different angle, then it's very interesting. So especially, for example, in the psychiatric domain, it's for depression, we kind of nowadays I think we aggregate the different depression symptoms into one combined score, but putting them together, the original features into one model would be very relevant to get the better discovery based on the multivariate, many features measured on this individual. Really good way to boost the power.
Thank you. And it's a very naive question, but I still want to ask, what's your opinion on the clinical, the direct clinical application? What is, you covered a lot of different topics, what's the closest to clinical use?
At this specific point, so I feel we need to do more work on the genetic side, on the modeling side, in order to get to the clinical prediction. And so my biggest worry is that the companies that sort of a little bit prematurely start approaching people, offering them some services when they tell that it's basically possible to do prediction using the genetics data, without fully explaining that it gives you like 1% of the predictive power, right? So, like, yes, it is associated with, but it's still 1%. And people are not, I think, not particularly good at understanding the numbers when, like, okay, 1%, what does it mean? It has to be visual. It's like, if it's this kind of pie circle and it goes, okay, this is unknown and we can predict it is 1%, right? Then it will be a very different attitude.
Well, it depends on the final application. You mentioned that you can find some small, well, it will be small, but still a small cohort, and then you can assign specific treatment or whatever. So
I think it's not tomorrow. It's in some like years from now. But I think it's about detecting the groups that are at risk, so that they get some priority. For example, in cancer and in Alzheimer, I think it's closer than in psychiatry. We are moving towards a precision psychiatry domain, but it's basically detecting the group that is at high risk so they can get more attention from the healthcare specialists earlier.
It's the last question from me. So it's related to the first one about the factors behind this progress. So imagine that we have, we will have discussion in 10 years, and what will be the driving factors again for the next big step? Is it really possible to do this next step, or maybe you need 100 billions of images? So is it possible to improve this? I mean, not specific this, but in general.
I think, as you mentioned, there are three dimensions to it. So collect more as we do today, collect another more rich phenotypes, right? More features, and also do the better methods. So I think it's all, everything complements each other, and it definitely looks that we are on a very positive trend, especially in psychiatry. I think comparing to 20 years back when there was like a whole wave of studies that didn't replicate. Now everything replicates. So I think we are capturing some real biological phenomena with this data, but we just need to work it through and get to bigger samples, better techniques, and we will eventually be able to bring it back to clinics so that people actually benefit from this new knowledge generated.
So
you for the lecture. On one of the Manhattan plots, you have shown one gene had particularly high load score, this one, in the middle.
Mm-hmm.
Is it known anything about this gene? Was the story taken further?
Yes.
To analyze the locus? Can you comment about it, please?
Thank you so much for this question. So yes, I think it's actually the highest peak here is a major histocompatibility complex, right? The thing that is heavily involved in immunity, right, on chromosome 6. So, and it's an insanely complex from genetics. This locus has the largest LD structure, meaning that it's a very large complex. It's very hard to investigate it deeper. So, for example, in our analysis, we tend to exclude this locus, quite unfortunately, even though we know its role in schizophrenia. So the first, I think, it was really interesting to observe that the largest peak of schizophrenia is in MHC, because there are some theories behind schizophrenia, also like hypotheses about neurotransmission, and but the immune hypothesis in schizophrenia is sort of not, it wasn't the first one on people's list, but this is an empirical evidence that points towards the role of autoimmune and immunity in schizophrenia. So what people done afterwards, they really, I think they nailed down it to the complement C4. So even though there are many, many genes, I think up to maybe 100 genes in the MHC locus, I can't say for sure, but I think they sort of boil it down to the complement C4 factor, which has different isoforms, and it's the specific isoform that is kind of hyperactive in the synaptic pruning. So I think the working hypothesis, there is a very good paper about that specific. It's a lab work, wet lab work, where they go and study this particular region of the genome. And I think the conclusion is that basically one potential explanation would be that during neurodevelopment, so during the neurodevelopment, there is an excessive synaptic pruning, and there is a little bit too many wires cut in the brain that somehow is associated later on with bipolar. So there is a large story. Even though we know it can't explain everything, but this must be a contributing factor, and maybe that might help to sort of tease out a small proportion of the schizophrenia sample that have a more specific mechanism that we can treat in a different way.
I may just have a question. So you have very accurately avoided when you discussed the correlation between genetic loci and schizophrenic phenotypes, the cause and effect relationships, because obviously the population correlation studies cannot give you that. Do you know any studies which take another angle? For instance, pair analysis, where one gene in several families is analyzed for cost segregation in the trial. And is this approach still used in?
So, I'm not directly aware of these studies, but you're totally right that the causal, like this is more observational studies, right? We can look for association and correlations, but the causal mechanism is something that we don't directly work here. There are techniques called Mendelian randomization. I'm personally, so we didn't apply them here in this center, and I'm still, it's based on the same data, so we need to look exactly what data do we get from Mendelian randomization, but people commonly refer to it as a putative causal relationship. So I think it would be more empirically valid to look at either stem cells or what I mentioned we can do very soon in the MOBA sample. So there was, I need to scroll back many slides back, but we have in order the MOBA sample, which is a trio cohort. It's the largest trio cohort in the world, to the best of my knowledge. It's in total 280,000 kids and parents together, so around 800 trios. And there we can look for transmitted and non-transmitted alleles and see is there, to elaborate on this question further. In terms of the one thing we can do on statistical perspective is, I think if I go slightly back on this one, right? So it's still not about the causal in terms of like cause and effect that you would observe in the clinical trials. But another problem with GWAS studies is that we look at this association signal, and a lot it arises through LD. So you might have one causal variant somewhere in a chromosome, But then, because the whole chunks of chromosome are inherited together, it will be associated with all of the variants in the same region. So, but because we know the structure of the linkage disequilibrium, at least we can take this out. So that is something that I work as a statistician to model the effects of linkage disequilibrium and go down to this level of additive genetic effects so that we are not getting a picture that is completely blurry because of the linkage disequilibrium structure. But this is not, so we call it a causal level or whatever, but it's just so taking LD out of account. It's not the same causal relationship that we would observe in the clinical trials, double-blind studies.
So maybe another question? Okay. Thank you very much.